Hello and welcome to BMY 4302 Virology. This course is brought to you by the Department of Microbiology, Faculty of Biotechnology and Biomolecular Sciences, University Putra, Malaysia. I'm Dr. Elisha. In the previous lectures, we have been talking about the Negative-Strand RNA virus, where we specifically look at the poliovirus, and we know that the poliovirus has the RNA genome with positive strain, whereby this RNA genome can be used directly for the productions of viral protein. And specifically, when we look at the RNA of the poliovirus, we notice that there is no cap. And in order to overcome this, the poliovirus have to have IRAS in their genome, whereby this IRAS is the site where the ribosome, the EIF, binds to allow viral protein synthesis. Then we look at the second part of the RNA virus, which is the envelope virus. So in that, we use dengue virus as an example for us to learn a more complicated from a naked virus. Then we learn a more complicated virus where the virus has an envelope surrounding the capsid now. So in dengue virus, unlike poliovirus, where poliovirus doesn't have a cap, but dengue virus do have a cap. So it basically just use exactly the same translation procedure as the host, where it has the EIF4E that binds to the cap and then binds again to the 4G and so on and so forth. However, we also notice that the dengue virus RNA doesn't have a poly-A tail, which is a site that is very important for the poly-A binding protein to bind to and circularize the RNA for efficient translations. So then we notice that there is some A-rich regions that spanning across the pseudoknot of 1 and 2 for the dengue virus at the 3' end, which allow the poly-A binding protein to bind to and then circularize, then the whole translation process going. Alright, so now that we have learned about positive strain RNA virus, we are going to make it a little bit more complicated now by looking at the negative strain RNA. So unlike poliovirus and dengue virus, the RNA can be used directly to synthesize the protein. Negative strain RNA cannot. So let's look at how this is being done. So the outline of these lectures, we will start with some introductions about the negative strain RNA virus, and specifically for this week we are going to look at the non-segmented RNA virus, where the virus has the RNA that is not segmented into a few segments but a long RNA. Then followed by next week we are going to look at the segmented RNA virus. Alright. As the introductions, there are a total of 29 families that has negative strain RNA, where 15 of the families are non-segmented and 14 of them are segmented. Let's have a look at the, this is the classifications that done by International Committee on Taxonomy of Virus, in short ICTV. So if you are interested in this particular website or committee, what you can do is just type ICTV space virus, then you click enter, then you'll come up with the link, then you can select them and you go to the website. And so this committee is the one that's responsible for our classifications and do all the taxonomy of the viruses so far. Now, when I was a student, the highest classifications for virus is just order. And we know classifications for life, the smallest unit will be the species, then genus, then family, order, class, phylum, and kingdom. However, for virus, during my time, it's just order. And now it's 2020. And you can see that the classification of virus has gone all the way to kingdom and even realm, right? So this is the classifications for negative-strand RNA virus. For RNA virus, they are under the realm of Riboviria, then kingdom of auto-RNA-Viridae. Under the negative-strand RNA is fall under the phylum of Negative-RNA Viridicotta. Okay, Negative-RNA Viridicotta. This is not M, this is RNA. Negative-RNA Viridicotta. Then we have two subfamily. One subfamily, Haploviricotina, which is the non-segmented virus, and we also have Polyploviricotina. So haplo, as you learned from science previously, haplo is single, polyplo will be many. So means single, non-segmented, polyplo, segmented. So over here, you can see that they have 4 classes and 2 classes, and I've clicked this, and now you can see in total the Haploviricotina. What we can see here is you have a total of 15 family, 1 family, 1, 11, and 1. 11 plus 4. 15 family. And for Polyploviricotta, we have 12 plus 2, 14 family. So in total, we have 29 families that is having a negative-strand RNA genome. So of these 29, 8 families are very important that cause human epidemics or outbreaks in the human history. And even now, we also suffer from some of the diseases. For example, the flu just come back all the times. And in the old time, where vaccine is still not available, the mumps and measles are also one of the some of the diseases that cause the outbreak in human history. Then we also have rabies that's transmitted by dogs, encephalitis, hemorrhagic fever. These are all the diseases that is very, very devastating and kills the patients. So what are the 8 families that listed just now? So we have Rhabdoviridae, Filoviridae, Paramyxoviridae, Pneumoviridae, Bonaviridae, which are non-segmented. Then we have the segmented Automyxoviridae, Bunyaviridae, and Arenaviridae. So for Rhabdoviridae, one of the examples that fall into this group will be the Lassa virus, the virus that causes rabies. So this is being transmitted by the animals that carry the virus and bite the patients or bite humans, and then the saliva that contains the virus will then be transmitted into the humans. All right. Malaysia used to be a rabies-free country for many years. Because of the vaccinations that implement in our country, and because there are some cases of smuggling some unvaccinated animals from neighboring country to Malaysia, and that causes the outbreak. Not really an outbreak. There's the first case that reported in Malaysia, and we lost our rabies-free country status. And it could take a few years for the country to declare again being a rabies-free country. Then for Filoviridae, we have Marburg virus and Ebola virus, and I'm sure all of us heard about Ebola virus, and this is one of the very dangerous virus that transmitted through direct contact, as missing here, direct contact with the patient's body fluids, like even vomit, the feces, all this blood, all these are being transmitted through Ebola. Can be transmit the Ebola virus to the next susceptible human. The death rate for Ebola virus is really high. Luckily, it is not airborne. Okay, right. Then we have the next one, which is the Paramyxoviridae. We have mumps and measles, which are well controlled at the moment. because the vaccinations that is being made compulsory or by the governments in Malaysia. So we have MMR, mumps, measles and rubella. This is the vaccines that come 3 in 1 that being given to the newborn baby and as well as along teenage age that you get this MMR. The virus is transmitted airborne, which is dangerous. So with this anti-vaxxer movement, anti-vaccine movement, like currently like spread through the world, we can see the mumps and measles coming back in several regions of the world, and which is quite dangerous. Alright. Then the next family, Pneumoviridae, we have one of a good example is human respiratory syncytial virus. RSV is a virus that has that is specifically infect children that is really young, and it causes respiratory disease in these patients. There's no vaccine available. So far, no successful vaccine has been developed for this virus. Borna virus or Pneumoviridae is a virus that infects the nervous system and cause neurological disease. And so far, there is no death case reported for this virus yet. Then move on to the segmented virus. So segmented RNA virus, we have Paramyxoviridae, and all of us are not this virus is not strange to us anymore, because this the flu virus that come back every year with some mutations, and you need to get new vaccinations every year to protect yourself from the virus. Then we have Pneumoviridae, where the Pneumonia virus, these are the virus that cause hemorrhagic fevers, and it's quite dangerous. Transmitted by mosquitoes and so on. Then we have Arenavirus. Arenavirus, the very good example of this virus is lymphocytic chondrial meningitis virus. So this is a virus that attack the meninges layer or the membrane that surrounded your spinal cord, and it's one of very dangerous virus that can transmit by urine or saliva. Alright, so these are some examples of the viruses which has negative-strand RNA that cause disease in human. So generally, the virus are enveloped. All the negative-strand RNA virus are enveloped. Okay, you will never see a naked negative-strand RNA virus at the moment. Maybe, who knows, one day in the future, you may discover one naked negative-strand RNA virus. We never know. Okay, because virology is an evolving course, that you get new and more new viruses that coming up, or we manage to discover more viruses with the current science and technology. Alright, so the virus has a helical nucleocapsid. What it means is that the RNA is always surrounded by nucleocapsid. Unlike poliovirus and dengue virus, they have the icosahedral capsid that's protect the RNA, which is located at the core of the capsid. The RNA for negative-strand RNA virus is always protected by NP protein or nucleocapsid protein. This nucleocapsid protein will bind to the RNA, as what we can see here. This is the RNA, will bind to the RNA and surrounded the RNA from very first nucleotide all the way to the end, and they form in a helical shape inside the virus. Alright, so this is what it means by helical nucleocapsid. So helical in like a spring shape inside the virus. So the RNA genome will never be released. Why? Because in our body, we will never have negative-strand RNA. Alright, so when there's a negative-strand RNA that occurs, the body will see as mutations or a foreign. So that's why the RNAs in your body will start attacking the negative-strand RNA. So in order for the virus to be successfully infect the cell, their RNA will always be protected by the nuclear capsid. They will never release free. And because their RNA is negative-strand, so this RNA cannot be used directly as if it is an mRNA or functional mRNA. So for the virus, they must always carry their own enzyme. Right, we have DNA-dependent RNA polymerase. So we have the RNA polymerase that read DNA as template to produce RNA, but we do not have RNA-dependent RNA polymerase. All right. So the virus must carry their own RNA. Okay. All right. So we'll look at it in more detail when we take one of the virus as example to study in details. All right. Then different family encode one or two glycoproteins. As what we can see from here, this is virus. So some of virus, they have just glycoprotein or just one G protein. As in this case, they only have one glycoprotein on the surface. But some viruses, they also have more than one glycoprotein on their surface. For example, in this virus, we have HN protein and F protein. HN and F. So hemagglutinin neuraminidase protein, and F stands for fusion protein. So all HA and NA, which is something similar to the flu virus, where they have two protein on their surface. So this is the genome to show you that the genes are being encoded by the RNA genome for these viruses. And please take note that this dotted line simply means that they are connected, they are not broken. Whereas for segmented, you can see this is one segment, this is second segment, this is the third segment, one segment, two segments. There are 8 segments here. So there's no connections, no connection, no connection, no dotted line. Right? So what we can see here is the arrangement for the RNA genome is quite conserved across the negative-strand RNA virus. So what we can see here from the front or the 3' end, we always have the nucleocapsid gene, where you can see the nucleocapsid, antiprotein, phosphoprotein, and the matrix. And in the middle, we have the glycoprotein. So you see there's a G, the HN and F, which is then glycoprotein. Then we have HA and NA. So this is what means in the previous lectures. Some have 1, some have 2, depending on the virus. And you will notice that most of the time, the polymerase gene will always be at the last. So for example, poliovirus. Poliovirus, the polymerase, if you remember, poliovirus genome separated to P1, P2, and P3. And P1 is the capsid, then you have P2 and P3, which is non-structural protein, and 3 have 3A, 3B, 3C, and 3D, and the polymerase is the last one, which is 3D. The same goes for dengue virus. So dengue virus also have the non-structural proteins that has polymerase, which fall at NS5, which is the last. And you notice that RNA virus, negative-strand RNA virus, also have their polymerase at the very last of their genome. This is why the reason behind this is because the polymerase that required for the replications of the virus is you only need very minute amount of that protein. So that's why it falls at the back of the genome, where you only maybe need 1, 2 of the polymerase. Then the enzyme is successful, is enough for the replications of the virus. Whereas for the rest, like nucleocapsid, you need a lot of nucleocapsid to protect the RNA. You need this fusion protein to be displayed on the surface of the virus. That's why you need a lot of this protein, whereas polymerase, you just need 1, 2. That's why most of the time the polymerase of viruses will fall at the end of their genome. Alright, so I'm sure you're not, you are quite familiar with phylogenetic trees, and we also mentioned previously, because of the technologies that we have now, that you can analyze the genome of the virus, where over here you can maybe choose the polymerase across the family of the viruses, and you just align them and try to compare them, and from there you will be able to build a phylogenetic tree, which will then group the virus accordingly. And you will notice not only you can use the polymerase, you can also use the nucleocapsid protein, or other proteins that is available. Then you'll be able to build this phylogenetic tree. So as an overview for this, here we separated the RNA virus into two, the haploviricotina and polyploviricotina, which is the non-segmented and segmented. And here you can see there are two orders here, and Negative-Strand where the Paramyxoviridae, the flu virus located, Paramyxoviridae, and then Negative-Strand. The one that we are very interested in, because there's too many viruses to study, the one that we are interested in is the Paramyxoviridae. So under the Paramyxoviridae, what we can see here, this is the figure that I took from SCTV again. So here we can see that Paramyxoviridae, it has 4 subfamily, and these are the subfamily that we can see under this family: the Avilaviridae, Metaparamyxoviridae, Autoparamyxoviridae, and Rubulaviridae. We also have some unassigned, means that the scientists still do not know how to classify these. So they belong to Paramyxoviridae, but still not sure how to classify them whether into this subfamily or just leave it as it is at the moment. So in each of the subfamily, we have genus of Meta-avilavirus, Auto-avilavirus, Para-avilavirus, and so on and so forth, what you can see in this whole list, right? So the one that we interested in is Autoavilavirus, where this is the virus that the Newcastle disease virus is being classified in. The virus that we are interested in is this NDV. So for microbiology students, you are not very, I'm sure this virus is not a strange virus to you anymore, because in the practical class you learn how to propagate the virus, and then you also do some extra experiments to analyze the virus, the presence of virus, for example using hemagglutinin assay, HA assay, and then you also look at the HI assay for this. Alright, so we always say the virus belongs to is also called the avian paramyxo serotype 1, because back in the old time there are a lot of avian paramyxovirus. For NDV, it's being classified as serotype 1. Alright, so the virus is first isolated in 1926 in Newcastle upon Tyne. Like I mentioned in the previous lectures, that in the old time when we name the virus, we always name the virus based on where the virus is first being isolated. So in this case, Newcastle upon Tyne is where the virus is being located. So it's called Newcastle disease virus. However, science is evolving, so people are improving, and now we try not to label the virus according to the location where it's being isolated. This is simply because, we try not to create some some unwanted cases, like for example the recent outbreak or pandemic worldwide, which is the coronavirus. The virus that caused this COVID-19 is COVID-19 is the disease. Okay, the pandemic is called COVID-19, but the virus that caused COVID-19 is called SARS-CoV-2. Okay, what it means is abbreviated for severe acute respiratory syndrome coronavirus 2. All right. I know some countries are still naming the virus as Wuhan virus because the virus is isolated from Wuhan, was first isolated in Wuhan. But since the WHO already named the virus as SARS-CoV-2, we might want to stick to the virus, the proper naming of the virus. Okay, so 1926 in Newcastle upon Tyne in England, and the following year it was isolated in island of Java in Indonesia. Okay, this is 1926, so don't imagine it as the virus traveled through airplane all the way to Indonesia and transmitted there. Alright, so Newcastle disease virus is sort of like endemic throughout the tropical country. For example, in Malaysia, in Indonesia, we are a country that is very susceptible to Newcastle disease virus. And because of the advancements of the science, and now we found a vaccine that can be used that protect the chickens from being infected by Newcastle disease virus. Now the virus can infect more than 27 orders of birds, so there are a total of like maybe 52 birds. So it can infect like more than 50% of the birds order, including chicken. So just imagine if there's an outbreak in Malaysia and in the poultry farm, you will not have any chicken to eat. Okay. So the virus can be separated into 3 pathotypes: lantogenic, mesogenic, and velogenic. The lantogenic is the mildest of the 3 pathotypes, where the chicken might be infected by the lantogenic strain, but they do not show any symptoms. It could be asymptomatic, or the virus cause only very mild flu symptom. So it will just go away and it will not cause any death to the chicken. Then move on to the second one, which is the mesogenic. The mesogenic strain is the strain that can cause up to 50% death to the chicken populations. For example, a poultry farm that has 1,000 chickens in it, and maybe it will kill half of it, like 500 chickens in the poultry farm. The virus, this particular pathotype, will cause inflammations on the neurological system, cause disease in neurological nervous system. Sorry. Then the last one, the velogenic. This is the most severe one that cause 2 types. One called visceral trophic, the other one called neurological trophic. So what does it mean is that visceral trophic, the chickens that is visceral trophic velogenic, it will infect the gastrointestinal tract, whereas the virus that causes neurological trophic, it will infect the nervous system. And what you can see here, these are the chickens that have been infected by Newcastle disease virus, and you can see most of the chickens will be very tired. They will just want to sit down, they close their eyes, they cannot control. And if you have seen some video, just type Newcastle disease virus in YouTube, and you can see some of the chickens have their head twisted at the back as they walk, and they just cannot control because the virus attacked their nervous system, and they just cannot control their body movement. So the lantogenic, because it's mild and asymptomatic and causes asymptomatic in the chicken, this particular pathotype has been used as a vaccine to protect against the more severe virus. Alright, so let's move on. Now let's look at the virus in detail. So what we can see here is a Newcastle disease virus that I drew when I did my master's degree. And the virus is quite devastating, and because of that, I'm trying to make the virus more lively. It looks like a sunflower. Right. So in the core of the virus, what we can see is the RNA that is surrounded by nucleocapsid protein. So the RNA is always inside the nucleocapsid, and it's being protected by the nucleocapsid protein or NP protein. And you can also see two other proteins that is touching the NP protein, the pink color and the red color. So the pink color is the phosphor protein. So phosphor protein is also important in the replications of the virus where it activates the polymerase. The red color is the L protein or long protein. So this L protein is actually the polymerase of the NDV. So together, these three is responsible for the transcriptions of the RNA. Alright, moving upward, you can see the blue color, or if you can see here, this is easier to see. Here you have the nucleocapsid, and then outside, underneath the membrane layer, you have a layer of blue color here, which is the matrix protein, the M protein. Alright, the matrix protein is highly hydrophobic, so it normally lies underneath the membrane, and this is a protein that is responsible for the budding of the virus during the assembly. Alright, so we will look at that during the virus life cycle. And moving out, you have a layer of membrane where this membrane derived from the host cell membrane. So this is the host cell membrane, and on the surface you have two proteins. One is hemagglutinin neuraminidase protein. This is the proteins that play a role in the attachment of virus to the host cell. And we also have another protein, which is the fusion protein, the F protein fusion. So the F protein is responsible for the fusion of the virus into the cell. Right? So this is the structural proteins that is present on the virus. So let's look into detail of this virus. The virus has a spherical pleomorphic shape, meaning that the virus can be spherical and it can be elongated like a filament and so on and so forth. That's why you can see the diameter for the size can be from 100 to 500 nanometers. So most of the time they are spherical shape, alright, for NDV. And the RNA is non-segmented, single-stranded RNA polarity. And what we notice is that the RNA, the total size of the RNA can be from 15,186 nucleotide, 15,192, or 15,198 nucleotides. And if you look carefully, you will notice that all this can be divided by 6. So this is a very important characteristic for Paramyxoviridae or for NDV, right? The nucleotide will always be able to be divided by 6 simply because 6 nucleotides will be bound by 1 NP protein. Alright? I mentioned earlier on that the RNA is protected by nucleocapsid protein, and 6 nucleotides will be bound by 1 NP protein. Alright? So because the RNA is negative strain and it is foreign to your body, and the RNAs will be happily to digest the RNA, so it must be protected. In order for these protections to be efficient, 6 nucleotides must be bound by 1 NP protein. Alright? And you cannot have extra RNA towards the end, or not enough. In order for the NP to bind efficiently, it must have 6. Alright? So this is what it means by rules of 6. So that's it. That's the end for part 1 for the new envelope Newcastle and negative-strand RNA virus. And in the next lecture, we are going to talk about the life cycle of Newcastle disease virus. Thank you.